Hello, my name is Carmen Sanchez-Reddick, and today I will be discussing the differences in the morphology and reproduction of Boltenia villosa across a latitudinal gradient. Boltenia villosa is a member of the class of ascidians. Ascidians are sessile, solitary, or colonial filter-feeding marine invertebrates, also known as sea squirts. Despite their unassuming nature, ascidians have proven their influence in various fields of study, including genetics, medicine, ecology, and the study of marine environmental issues. Sea squirts hold tremendous value, which underscores the need for researchers to advance existing literature for ascidians. Yet these advancements require the biology of the members of the class of ascidians to be well understood, and that remains a continuous goal. One species in such need is Boltenia villosa. From the pigment of its larval muscle cells to settlement preferences, the timeline of metamorphosis, the larval and settlement stages of this species are well documented, but little is known about its adults. Outside of the current literature, scientists of the Pacific coast have noticed trends in the distribution of Boltenia villosa, but these observations have not been quantitatively researched. Early documentation of Boltenia villosa describes a relationship between the body size and the stalk length, as individuals with smaller bodies tend to have longer stalks, and individuals with larger bodies tend to possess very short stalks, as seen in Figure 1. Anecdotal evidence suggests that larger individuals with shorter stalks make up the population found in Washington, while Oregon populations consist of smaller individuals with longer stalks. The present study aims to develop quantitative understanding of how morphology as well as reproductive output changes across the latitudinal gradient of Boltenia villosa, and to uncover possible cryptic species through molecular analysis. Boltenia villosa samples were collected from a shallow water site in the San Juan Islands and a deep water site off the southern coast of Oregon during the fall. To quantify any morphological differences, a combination of morphometrics and scanning electron microscopy were employed. The external anatomy of all 212 Boltenia villosa specimens were measured using calipers, with measurements of the body length and width and stalk length and width being collected. SEM, or scanning electron microscopy, was used on the spines covering the tunic of the species to photograph any microscopic differences in the spine character. To see if there were also any reproductive differences, gonadosomatic indexes, or GSIs, and histology were done. GSI involved weighing all the internal anatomy of an individual, then dissecting and weighing their gonads to see how much of an individual's internal anatomy was used for reproduction. Histology of gonads allowed for the diameter of the first 100 eggs, or also called oocytes, to be measured from several Oregon and Washington specimens. Finally, molecular analysis was done for 5 Oregon specimens to be compared against existing Boltenia specimens from Washington and British Columbia found in Gene Bank. Looking towards Figure 3, a multidimensional scaling plot showed a clear separation between the two populations with very little crossover, as the 4 variables of stalk width and length and body width and length were looked at for each individual. There was also microscopic dissimilarities in the spines that cover the tunic of the species, looking at Figure 4. SEM revealed the presence of tiny spines or protrusions from the main spine on all but 2 of the spines collected from the Washington specimens. In the Oregon specimens, their spines were smooth, except for the case of 1 sample. For GSI, looking at a 2-tailed, 2-sample t-test, it was determined there was no significant difference between the populations regarding the percentage of body weight that can be accounted for by the 2 gonads of any one individual. While the GSI values did not differ greatly, the diameters of the first 100 eggs in individuals from Oregon and Washington were. First, looking at Figure 5, you can see how I counted and measured 100 oocytes, with the purple dot being the center of the nucleus and the white indicating what was measured. Figure 6 shows a box and whiskers plot for the diameter of the eggs or oocytes. On average, individuals from Washington had larger oocytes than individuals found in Oregon. But the Washington specimens also showed a much higher degree of variance in oocyte diameters than those in Oregon. This indicates that individuals in Washington dedicate more energy to egg production than Oregon individuals. When compared to Boltenia villosa, specimens analyzed from Washington and British Columbia, there were over 98% molecular-based similarities. This indicates that the populations from Washington and Oregon are the same species, and that their morphological and reproductive differences are not a result of interspecies differences. Despite their genetic similarities, there are two distinct morphotypes with significantly different reproductive outputs found between the Anacortes Washington and Cape Arago, Oregon populations of Boltenia villosa. Individuals in Anacortes could be identified by their large bodies, short stalks, and hairy spines, and their larger oocyte size, while the Cape Arago population consisted of individuals with small bodies, long stalks, smooth spines, and smaller oocytes. The difference in the proportion of gonad to total body weight was not significant, GSI, which may be explained by members of the species always maximizing their fitness and reaching a similar GSI proportional limit no matter their population. These results create a strong foundation for continued exploration into these populations of Boltenia villosa and the possible explanations for their differences. These differences, of course, being seen in Figure 7. Future research may examine the impacts of food availability and water temperatures on oocyte diameter or the influence of water movement on a given habitat's morphotype. Either way, without the present study's research, no future research could be conducted. These are my references, and of course, I would love to acknowledge the Clark Honors College Mentored Research Program for funding portions of this research, and my mentor, Dr. Craig Young, and the entire Young Lab for sharing their knowledge, guidance, and support throughout this entire process. Thank you very much.